And so with that, I'll introduce our first speaker, Eric Bridgeford. And so Eric is a graduate researcher in the Department of Biostatistics at Johns Hopkins University in the lab of Joshua Vogelstein. His talk is on batch effects or causal effects applications in human functional connectomes. And with that, I'll let Eric start.
Great, thanks. I appreciate the introduction, Stephen. So to start us off, I will discuss a little example here. So let's say we have n participants and we measure functional connectivity. Let's say pairwise correlation between the ROI time series in this case. My goal is to effectively address whether or not, at specific edges, is there a difference between the FC of males and the FC of females? So let's say I just do an associational analysis, maybe a linear regression at each edge, and I identify which edges are significant after some sort of an adjustment for the many comparisons that I have here, like Bonferroni correction. I find I have 99 edges that are significant in FC after multiple comparisons, and I go in and report my finding and ship my paper off to bioRxiv. Now some other investigator comes in, and let's say he gets a hold of a new group of people, takes a look at the data, and finds a new subset of edges. And kind of the problem that arises is that some of the edges are similar and agree between the two analyses, but a lot of the edges are a lot different than the ones I had originally found. And so sort of the question is, which edges are the actual signal edges? What went wrong in this case? And so in this study, there are effectively two problems with making this comparison. So the first problem is when somebody tells you, that I have a difference between two data sets, your mind should immediately jump to, tell me something about the study population. Were the study populations similar? How did the study population differ? And the problem with the local associational study is that we're not really seeing the full picture here. My inference is going to apply well enough to a population identical to the population I actually, measured FC for. But then when I try to go and generalize that to, whatever my target population is, or a new target population, that becomes a lot harder. And so basically the problem here was in data sets 1 and data set 2, the issue that we had was that the two data sets differed, by an order of magnitude, basically, in the age, sex, and racial composition of the individuals, as well as a number of other factors, which we basically term our signal-producing covariates. And so in this case in particular, data sets 1 and data sets 2, as you can see, the sort of age regions of the individuals, or the age ranges, are just totally disparate between the two data sets. And I can subvert this problem somewhat by doing things like, quote unquote, adjusting for age and sex. But the problem I run into there is that can end up basically aliasing the signal of interest that you actually want to measure, which would be the actual batch-related effect. And so what we do in a few slides is we'll actually just try to restrict ourselves to the shared regions of the covariate distributions in which the two data sets are basically similar enough in these sorts of signals of interest. And let's say now we have a sort of a third study where the experimenter realizes this issue. He goes to a new group of people who are effectively identical in covariate distribution to the people in data set 1. We'll call that data set 0. And he measures FC again, but he's at a different university. So and this time we end up with a much more similar agreement, but we're still getting disagreement in about 20% of the edges. And so now the actual statement that you start to be able to make is that you have a piece of evidence that the batch, the factors that relate to the site, the MR machine, the experimenter, the protocol, et cetera, is leading to this disparity in the detected signal. Kind of stated another way, we start to be able to have some piece of evidence that something related to the batch is what's actually causing the signal disparity between the two data sets. So to put this into practice, these sort of two pieces, we're going to look at the prominence of batch effects compared to local associational effects in the core metadata set. So for those who aren't familiar, the core metadata set comprises a few thousand individuals measured at a couple dozen of sites. And we basically restricted ourselves to the subset of data sets where we had a bunch of phenotypic covariates such as the sex, age, and continent of the individuals who were measured. So we estimated FC using the AAL parcellation. And our procedure was pretty straightforward. So first we compare the association between the dataset identifier with functional connectivity for two pairs of data sets. And next what we did was we estimated a causal effect between the dataset identifier with functional connectivity if such a comparison was possible. And so, by possible in this case, what I really mean is if the two data sets had sufficient overlap in the covariate distribution, very similar to what we saw with dataset 0 and dataset 1 in that mini example, basically after removal of individuals who are outside of the region of overlap in the covariate distribution. So in this first figure, we took a look at whether or not associational or batch effects were actually present in the data set. So we have a total of 276 possible comparisons amongst all of these 24 times 23 pairs of data sets where we removed duplicate comparisons since our test statistic in this case was symmetric. So we had 170 pairs that differed pretty substantially in the covariate distribution where we did not think such a comparison was warranted. Either this was, the covariate distributions didn't overlap whatsoever, or there were only a very small subset of the individuals in that sort of shared region of the two covariate distributions. And second, we had 106 pairs of data sets where we had a very similar covariate distribution. And those are shown on the right. And so effectively in both plots, what we ended up seeing was that, sort of associational effects between the data sets, as well as, effects where we can begin to make a more causal statement about batch effect, were both present and extremely prominent in the data set. So in the next figure, what we were trying to do was, how can we remove this sort of batch-related alias in the data set? And so we used a strategy called combat correction. So basically what happens with combat correction is across all the features, you fit an empirical Bayes model to identify components of signal that are specific to each batch. And you try to take that out. And so, what we can see in both of these figures is that, if we compare this to the previous figure, practically none of the effects are significant in this case after adjustment, which, it indicates we've done a pretty good job of removing, whatever batch-related signal we were detecting previously. So our next question was a little bit more high level. So on one hand, we've removed our ability to detect a batch effect. We don't really know what happened to the rest of the signal that we might want to investigate in our neuroscience investigation or whatever. And so the problem basically boiled down to the fact that, whenever you remove something, you're not really sure whether or not you're also removing other useful things that, might either alias or might be scaled in with whatever your batch removal strategy was. And so to kind of quantify how prominent this was, We identified a few signals of interest that we considered to be, sort of ubiquitous in that they're pretty often or nearly always present, but were unknown to the combat algorithm at the time it was applied. So to start off with, we took a look at a few between individual comparisons that we might be interested in making in Connectomes. So before doing combat, while we couldn't compare two data sets due to the problem that we mentioned at the beginning, we could still do inter-individual comparisons within a single data set and look for associational effects. And so what we did here was we took a look at whether or not sex and age effects were present before and after combat. And we found that after adjusting for multiple comparisons, both sex and age effects were fairly prominent. So we haven't removed this sort of inter-individual signal. The second question was, there are also a bunch of within individual properties of the connectome that we're interested in. So here we were seeking to check that properties that were present at the individual level before and after combat were still present. So our individual level signals in this case were the homophilic and the homotopic differences in connectivity. The homophilic difference is the difference between edges in the same hemisphere and the opposite hemisphere. This effect doesn't tend to be super prominent. And on the right plot, we can see what happens with the homotopic difference in connectivity, which was the difference in connectivity between edges that are bilateral homologues, for example, region A in the left and right hemisphere, and edges that were not bilateral homologues, region A and region B, irrespective of hemisphere. And this tends to be one of the strongest signals in functional connectivity that we tend to find, in that it's present, I think 100% of the time across all of the connectomes in the core data set. And so the conclusion here was that combated connectomes tended to yield the same inference as the raw connectomes at the individual level, which was not the case with a couple of other fairly naive methods for batch effect correction that are used fairly prominently in like the genomics literature, like z-scoring, ranking features within a data set, things like that. And so in summary, we proposed a strategy for basically more precisely posing the batch effects problem as a causal problem. We proposed a method for estimating batch effects and differentiating batch effects from associational effects at the interdata set level. We showed that batch effects could be successfully removed with combat correction, and we showed that combated connectomes preserved between and within the individual properties of interest. So finally, some people I'd like to thank are my PIs, Josh Vogelstein and Brian Caffo, a few colleagues that I work extremely frequently with, Mike Powell, Anton Eliakin, and Noam Finkelstein, and finally, the Consortium for Reliability and Reproducibility. Thanks, guys.
Thanks, Eric. That was really exciting. So yeah, just a reminder, if you want to ask any questions, feel free to ask in the Q&A. Just have a few minutes for those. I'll jump in real quick. So you talked about looking at covariates such as age and sex. Are there other covariates that you're interested in looking at? Particularly, what comes to mind for me is like different scanners or something. Are there, variations across the measurement devices themselves that you might want to correct for?
Yeah, no, I think that's a great point. And so when I say batch in this case, that's kind of one of the signals that we would be interested in. Mm-hmm. In summarizing with some sort of a batch effect would be things like, if I measure person A at MR machine 1 and 2, and MR machine 1 and 2 are totally different models, that would be one of the batch related effects that I would kind of want to pick up, mm-hmm. With however I was measuring batch. Within the core data set, what tends to be the case is I think the overwhelming majority of the data sets are collected on similar MR machines in that I think most of them were Siemens and Philips, as well as using very similar scanning parameters. So that kind of made the fact that the batch effect was, I think it was present like 85% of the time. I haven't specifically looked at how it differs when you're looking at identical MR machine and identical scan parameters versus otherwise. But there's definitely a lot of examples where it was, same MR machine, same exact scanning parameters, still batch effect present. So
yeah, that seems to be a thing for a lot of other neuroimaging recording modalities as well.
Yeah,
Awesome.
absolutely.
Great. Well, thanks again, Eric.
Great, thanks.
So yeah, I'll introduce our second speaker.